owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining us from today. I pay my respects to the elders, past, present, and emerging, and to the Aboriginal elders of other communities. My name is Sepide Walimer. I'm a postdoctoral research fellow at CCE-MMP from Melbourne University Node. I will be chairing today's seminar. I just go through some housekeeping rules just to let you know that this meeting will be recorded and it will be available in CCE-MMP website. Please stay muted throughout the presentation, but there will be a Q&A session at the end of the seminar, which you can unmute yourself and ask your questions. And please feel free to type your questions in the chat as you think of it during the seminar. It is my pleasure to introduce today's speaker, Professor Stefan Raunser. Professor Raunser studied chemistry and biology at Johann Gutenberg University. He did his PhD in the group of Professor Werner Kulbrecht at the Max Planck Institute of Biophysics in Frankfurt and received his PhD in biochemistry at Johann Wolfgang Goethe University in 2004. Professor Raunser is a structural biologist whose research focuses on understanding molecular mechanism underlying cellular processes in the healthy and diseased organism. He is director of the Department of Structural Biology at the Max Planck Institute of Molecular Physiology, adjunct professor at Technical University Dortmund, and honorary professor at University of Duisburg-Essen. With his research group, he uses a multidisciplinary approach, including biochemical reconstitution, high-resolution cryo-EM, and electron cryotomography, primarily to investigate the structure of macromolecular complexes that play a crucial role in cell physiology, with a particular emphasis on toxin-mediated membrane permeation, the molecular details of muscle contraction, and the dynamics of the eukaryotic cytoskeleton. He's a scientific member of the Max Planck Society and an elected member of the North Rhine-Westphalian Academy of Sciences and Art, the German National Academy of Sciences, Leopoldina, and EMBO. So today he's going to talk about bringing life into frozen proteins to elucidate their molecular mechanism. So with that, I will hand over to Professor Raunser, and please start your talk whenever you are ready.
Thank you very much for this very kind introduction, and good afternoon to Australia. So I'm going to share my screen. Just a sec. Can you see my screen? Does it work? Okay, yeah. Great. So then I'll start. So because you are from the Center of Membrane Proteins, I concentrated the first part of my talk on our work on membrane proteins. And there we are very much interested in the pharmacological modulation of iron.
What time have you got it booked? What?
10.
Okay.
I'm pretty sure. Yeah.
Sorry, please everyone mute your microphone. Thank you.
Okay. I can.
Sorry about that.
No problem. Yes. And in particular, we use electron cryo microscopy as a tool for the structure-based drug development, as the title of this review here. Wait, now it's frozen somehow. Okay. So the main targets, there is another important aspect, is that we are interested in insecticides and how we can use channels as targets for insecticides. And there are several of them on the market. They mostly attack channels and transporters and other molecules in neurons, because by that they act very quickly on the insects, or as in this case, in muscles for the Ryanodine receptor. So this is something we have done early on, almost 10 years ago. We started with working on the Ryanodine receptor. Here, this is Ruslan's work, where we pioneered the reconstitution of this huge 2.2 megadalton membrane complex into lipid nanodiscs. You can see that here. And this allowed us to get a reconstruction of this huge protein, where only this part here sits in the membrane, and all the rest of it is there to modulate the channel which sits down there. So here you see this beautiful, huge structure. I won't go into detail because it's a relatively old study, but what we could show already then was when we compared structures of calcium-bound and calcium-free, there were conformational changes and also conformational changes itself in the molecule, depending just on the molecule itself. So we had a lot of fluctuations, and you see that these movements up there in these extra membrane domains resulted in movements at the center, at the gate region. And binding of different molecules to this to the extramembrane part results then in conformational changes inside the gate. Right, so this is the calcium binding site. Of course, at this time the resolution was not as high as we are used to it nowadays, but later structures confirmed that our model here that we put out was correct. So what we predicted based on these structures is that we have in the closed state without calcium bound, we have conformational changes that where the channel switches from open to close, and once calcium is bound, this puts the channel in a more active state so that it can translocate ions. But what we were, so Ruslan took this project with him when he became an independent group leader. He's now a professor at the Vrije University in Brussels. And we then continued mostly on trip channels and also on their regulation and interaction with pharmacological agents. So why a trip channel? So most of you know what they are, because the Nobel Prize, I think last year, was given for the discovery and the characterization. So there are 7 different families of trip channels, and you see all of them, and they have different functions. They're involved in thermosensitivity, nociceptions, and store-operated calcium entry. So what they have in common, they are all cation permeable channels. So some of them are for sodium and calcium, others are more specific towards calcium. So I have to open this. Nice. So we were very much interested in the TRPC channels and their role in calcium homeostasis. So this has been very controversially discussed in the past, and it depends very much on the cellular environment and the method of measurement. So there were several studies that show that TRP channels form receptor-operated calcium-permeable, non-selective cation channels, whereas others found that TRP channels are activated by calcium store depletion with moderate to high calcium permeabilities. In connection to that, TRP channels have been shown to directly interact with calcium, IP3 receptors, and calmodulin. STIM1, the lipid binding protein, STD1, the G protein, GIA2, and nitrogen oxide. So you see there are many interacting proteins, so that makes this channel very interesting to study and to see how it is modulated and regulated. It belongs to one of the 7 members. I told you that. calcium and sodium permeable. That you find them in many cell types and tissues. Importantly, they play a role in vascular relaxation and neurotransmitter release, and they contribute to axonal regeneration after nerve injury. And what's important for us, because we also work very much on cardiac tissue and muscles, there are necessary mediators of pathologic cardiac hypertrophy. Okay, so when we started with it, we did a classical approach. We looked at many different orthologs. In particular, we concentrated on the TRPC4 channel. It's 1 to 7 exists of these TRPCs. So we concentrated on TRPC4. That had the reason because a collaborator of us in the institute, Herbert Waldman, concentrated on this channel for drug discovery. So we looked then at different drip channels from organisms and tagged them at the end of the C-terminus, expressed them, and checked for the best one. So the best one we found was the one from zebrafish, and with this one we continued, solubilized it in DDM, CHS, purified it using a strap tag and size exclusion chromatography, and exchanged it afterwards to amphipods. And as you can see, this gave us a nice homogeneous distribution of particles that allowed us to then solve the structure by single particle electron microscopy. So this is Barati's work, and you nicely see that a relatively large part of the complex sits in the cytoplasm, and when you look from top, you see that it forms a tetramer. It's also interesting that there's always two helices of one subunit. They flip over to the adjacent protomer, so that it's really kind of stabilized. And this part here, the little part, when we go back once, we see that this is going into the center here, and this is forming the pore, which is actually the most important part, of course, of this trip channel. So when we look at the domain architecture, we see here a huge voltage sensing like domain. I'll show you why it's called like domain and not voltage sensing domain in a second. We have this pore domain and the trip domain that connects this part here by the rib helix to the long C-terminus. So the most part of the C-terminus here is 200 kilodaltons, is not resolved in none of the structures of the trip channels. And the other side, we see this the N-terminus, where we have 4 anchorin repeats, and they are connected by this helical linker domain. This one connects basically the pore domain, or like the membrane domain, with the cytosolic domain. So why is it not this voltage sensing domain, just a voltage sensing like domain? When we compare it here with the voltage gated potassium channel, we see that we have here mostly pairs of, here we have negative and positive charges on opposing sides here and they're separated by a hydrophobic center. And what happens during a voltage change is that basically it moves it flips from one place to the other, and this results in conformational changes. But in trip channels here in the TRPC4, ours from zebrafish, we have only one pair and this hydrophobic part, but the second pair of residues of electrostatic interacting residues is not there. So therefore, it's only a voltage sensing like domain by fold, but not by function. The drip channels are activated by calcium binding, and we saw here a density that would correspond to a calcium. Of course, the resolution was not high enough to 100% say that this is really calcium, but when we check the position and the residues, which are highly conserved, and compare it to a TRPM structure where the authors place the calcium. This is exactly the position where we would expect the calcium that activates the channel. When we look from the top, so we see where we enter the membrane domain, we see that it's highly negatively charged. And this seems to be important because if you mutate residues up here, like this glutamate, then the gating of the channel is much less efficient. So one seems to need a negatively charged sink, basically, for the ions to enter the channel. Here is the selectivity filter, which is kind of in the middle. So from the size, it's you see here from the radius, it's about 4 angstrom. Van der Waals radius. So that means that the ions need to be partially getting rid of the hydrogen shell, hydrated shell, so that they fit through. So it looks like they are partially hydrated only when they go through. Afterwards, when it goes to the selectivity filter here, we have a hydrophobic cavity. And underneath, it's clear at this gate, which we call the lower gate, that the channel is closed in our conformation. What's interesting is this ankyrin repeats that in most of the TRP channels of the other families, this is straight. In our case, it flips over and interacts with the protomer next to it. And this completely stabilizes this tetramer and also kind of closes the bottom very well of the channel. And another important thing that we found was that basically this coil domain, that's called coil domain because most of the structures it forms a coil at the bottom. In our case, it's basically just the straight 4 helices that come out of the channel. So it's not really coiled. But it's called domain. Yeah, there were many other things that are important, especially here. We have this conserved glycine in the N2 motif, as you can see here. And this is important to connect the TRP domain with the pore domain. And this you should keep in mind, because when you look at here, we have this pore domain. This is connected by the TRP domain to the periphery and also to the bottom here. So if we have changes that happen here at this position or there, they have a direct influence on the channel and on the gating of the channel. And therefore, this rib helix is kind of important when this is basically sitting parallel to the membrane and reaches out of the protein and goes back in here. So this loop that connects this part with the upper part has not been resolved in the structure. And we found lipids. That's also nice, but these were the lipids that we added during purification. It's phosphatidic acid and the CHS. So the CHS sits at this pocket over here, and up here sits the PA. And we think that these positions can likely be occupied by activating or inhibiting lipids in vivo, something like DHE, for example. So this was the picture before we started. It was a review by our colleague Christine Ziegler that was published early in 2018. I guess it was written in 2017. And there were all structures of the TRP family were solved, but not of the TRPC. And in the end of 2018, Stefan wrote a review because then the structural description of the TRP family of all TRP families was complete, and our structure appeared here, which made us very happy. But then later we got very much interested. The structure is nice, it explains the mechanism, but of course it's also interesting to know how you can manipulate the structure in order to cure diseases. And for that, we teamed up with Goldfinch Bio, a company in the US, and we tested the binding of pyridoxine-based inhibitors. They're all called like GFB for a Goldfinch Bio, and with these different numbers. And they have been shown to be inhibitors, especially this 8438 showed promise as a potential drug for the treatment of proteinuria kidney disease, exhibiting overall favorable in vitro and in vivo properties. So in this case, in vitro mouse podocytes were protected from protamine-induced injury when treated with this inhibitor. And to check where this class of inhibitors bind, we solved 3 different structures with 3 slightly modified inhibitors, and we found they all bind to the same site, which is the voltage sensing-like domain, but on the very bottom, where it can interact as well with the TRP domain. So you see it, they all fit nicely inside this pocket, and dependent on the changes, we have additional interactions with residues inside the pocket. So what we see here is that we have binding as a cartoon to this VSD. And what we propose is this stiffens up the interactions with the TRP domain here, resulting in a lockage basically of the channel into the inactivated conformation so that calcium ions cannot go through. In addition, we looked for calmodulin. So calmodulin has been shown to bind and regulate the TRP channel. At high calcium concentrations in the cytosol, calmodulin binds to its calcium bound state, and it inhibits calcium entry. At low concentrations, calmodulin changes its conformation and dissociates from the channel. And the store-operated calcium entry pathway hypothesis further proposes that calmodulin binds to the channel at resting state, prevents TRPC4 from being spontaneously activated by IP3 receptors. And then the calcium levels in the ER go up, but not in the cytosol. When they drop, the affinity of the IP3 receptor increases, and calmodulin is displaced through a conformational coupling mechanism. And this activates the TRP channel. So, long story short, we wanted to understand how calmodulin binds to the TRPC4 channel. And for that, we reconstituted the complex and found that we have up to four calmodulins binding to the TRP channel. So we had to find the classes where we have only one calmodulin bound to the tetramer. And this is what we then use for our reconstruction. And you can see here nicely that calmodulin binds at the periphery of the TRP channel. And what's nice is that this connection that I showed you before between the rib helix and this the TRP domain here is now resolved. So binding of calmodulin to this called curve domain down here results in an ordering of this region, right? And the stabilization in total of this connection between the TRP and the curve domain. What was interesting is that, and different from other channels, is that the calmodulin in our case binds to the periphery. So there have been also binding of calmodulin shown to helices inside isolated peptides from in here, because they have a calmodulin binding site. However, in our structure, this would be obstructed, the interaction, or like this site, this interaction site, by the C-terminal helices here. So, and therefore, in comparison to the others, calmodulin basically goes onto the bottom and blocks the channel. In our case, it sits on the periphery, and therefore we get a stoichiometry of a possible stoichiometry of 4 to 1 And in this case, it's always one calmodulin in one tetramer. So what is the effect of it? Again, it's similar to the inhibitor. The calcium binds and the calcium concentration increases inside to this peripheral region here, and this results in a stabilization of the closed conformation, and no calcium goes through. Now, of course, it would be nice to also have a structure of the open state, but I can tell you we have been very hardly, not hardly, it was very hard, and we have extensively been looking for an activated state, but so far we were not lucky. So let's hope that will happen soon. Another channel we were very much interested, also especially in regard of insecticide research, is the calcium activated potassium channel Slowpoke. And this was Toby's work here. This Slo are potassium channels with extraordinarily high conductance, and they're activated by membrane potential and calcium. They're involved in membrane potential repolarization, for example, in myocytes, and are important for muscle contraction, audition, neurotransmitter release. And of course, one can believe, when this Slo is deregulated, it causes muscle tremors and paralysis, also in insects and nematodes. So therefore, Slo is an interesting target for novel insecticides and antiparasitic drugs. So Toby solved the structure of calcium-bound and calcium-free Drosophila at 2.4 to 2.7 angstrom. It's a nice resolution that allows you to build a atomic model extremely well. And what you can see here, the build of these channels, is that we have the selectivity field in the center, a pore domain, a voltage sensing domain, so it's similar as before, but this time we have a huge gating ring here. And this binds up to 8 calcium ions. And when we look at it, what happens is when the calcium ions bind, this results in conformational changes that open the gate here and allow potassium to enter the selectivity pore and then gets to get to it. So let's look at the conformational change here. I see here this calcium gate. Calcium is binding, and this results in a conformational change. So the blobs here is a connection to the transmembrane region. And this basically pulls on these helices and results then in the opening of the gate, the lower gate of this channel region. So it was nice, and of course the thing was that this have already been known from mammalian channels, but we were interested in how this protein interacts with pharmacological agents. And to this end, we studied first verrucologen, which is a fungal toxin, and when you add this, the channel gets inactivated. So how does that work? And we will see that now in the structure that verrucologen binds up here, 4 of them. So for each protomer, like a subunit, we have 1 verrucologen bound, and it binds underneath the selectivity filter here. But what it does, it's not only sitting directly down here, but it's basically blocking the movement of this helix that is important for opening of the gate. See that in a second. So this would be normally the opening, right? And because verrucologen is sitting there, the channel cannot open anymore and stays in the inactivated state. In addition, because we have, this is an MD simulation showing, the accessibility, water accessibility of this region underneath the selectivity pore. And you see that this cavity is very electrophilic, and potassium can go in and be translocated through. In the case when verruculogen is bound, you see that because this molecule is highly hydrophobic, that it's very difficult for a potassium ion to enter this hydrophobic region here. So in addition to basically inhibiting the activating movement of these helices, it also provides a hydrophobic shell around the selectivity pore, selectivity filter, and therefore the potassium channels cannot go through. Yes, and then we checked another one. It's called amodepside. Many of you will know it, probably not directly, but this is a typical dewormer for dogs and cats. And what this amodepside does is basically it activates the slow channel, but in a way that it's not controlled anymore. So when you add this amodepside at high calcium concentration with the voltage, it's just independent on voltage, and the calcium we add, it's constantly activated. So why is that? This is kind of interesting. This molecule, you see, already has a hole inside, and it sits just as in one copy as a plug. It looks like a plug underneath the selectivity filter here, right, as one molecule. So it kind of really fits in, as I said, like a plug. But when you check it carefully from the bottom, you see there is space in the middle where a potassium ion could fit through. So what this plug does is it's keeping the channel in its activated state. However, it's not optimal because the potassium ions have to pass through the amodepside into the selectivity filter, and therefore the activation is partially hampered. So you see that here in the selectivity filter, and then we have an additional filter, so to speak, that sits underneath, and therefore the we have a suboptimal coordination of the whole thing, and it's activated but not as active as a fully wild-type activated potassium channel. So when we put them together, we see that it stabilizes the verruculogen stabilizes the closed state conformation and creates this hydrophobic environment in the cavity, whereas amodepside stabilizes the open gate conformation and adds a layer of suboptimal coordination to the selectivity filter. But I told you before about these insecticides, and this was also the reason why we used the Drosophila channel. And although they are quite similar to the human form, we compared them using this program BiteNet. BiteNet uses an object detection convolutional neural network to predict binding sites in protein structures. So you see many of them, they are found in the Drosophila and the human form. However, there were two regions, or one region, basically. This is this RCK2 pocket, as we called it, that did not appear in the human form. And there is this kind of interesting pocket that has never been targeted by a molecule. So this pocket, you can clearly see we have up here the conserved site that we find both in the human and the Drosophila, and down here the orange residues, they are different between Drosophila and human. So this site, because it's also different from the calcium bound to the calcium free state, could be optimal to develop an insecticide that keeps the channel either in the active or in the inactive state, and at the same time is has high affinity to Drosophila and not to human the slow channels. Another interesting one is, I told you, this is S6 pocket. This is found in humans and Drosophila. So So this is not a good candidate for insecticides. However, it fulfills all the prerequisites for a drug binding site, which is hydrophobic, and it only exists in one conformation. So if one had a molecule that bound in this cavity here, that would keep the molecule, the slow channel in a calcium-free state, that means an inactivated state, and could be used for the treatment of diseases potentially. Okay, so that was the first half of the talk. The second one, I want to concentrate a little bit on our studies on the TC toxins of Photorhabdus luminescens. So this is this protein Photorhabdus. It belongs to the same family as E. coli. It also looks similar, but what's interesting, it shows this bioluminescence. So it's the only known terrestrial bioluminescent bacterium, and it has an arsenal of insecticidal toxins. Therefore, we are interested in this organism, and especially in the toxin complexes called TC toxins. They're composed of 3 different components, A, B, and C, which assemble here before they enter the cell. It binds to the receptor on the surface, which results in endocytosis. And once it's endocytosed at the late endosome and the pH drops, the A component forms a pore through which then a part of the C component is translocated through. In our case, this is an ADP ribosyl transferase that modifies the actin cytoskeleton, and that results in an uncontrolled polymerization of the cytoskeleton and ultimately to cell death. So here you show you a short movie where the TC toxins are put on the cell, and in a very short time, the cells die because of the cytoskeleton collapses. So over the years, we have studied these complexes in different variations. So the holotoxin itself is 1.7 megadalton in size for this TC toxin for Photorhabdus luminescens. And we looked at the A components and the BC components, and also in complex. So this is how this complex looks like. It's the A component has this bell-shaped structure. When we look inside, we see that the pentamer that's forming here is surrounded by this shell, but at the inside we have this. I always call it vuvuzela-like structure, like a very long channel. When we cut deeper inside, we see that we have a long channel that looks a bit like the tip of a syringe as well. And how is this all built up? So we have this very large alpha helical domain that forms this shell around this central pore domain. We have a receptor binding domains that sit outside. So the neuraminidase-like domain in total, we have 4 of them, and plus the neuraminidase-like domains, that's basically 5 potential receptor binding domains. And the whole part here is connected by a very long linker to the pore forming domain and a TCB binding domain on top. This build is found in many different TCAs. So this is from different organisms. This is Photorhabdus luminescens, but we have also Yersinia pseudotuberculosis here, or Morganella morganii, and this is from Xenorhabdus. So overall they look very similar. What's different in them, you can see that at the periphery, some of them have a little bit more weight than others, is the amount of receptor binding domains at the periphery. So some of them lack 1 or 2 or even 3, and this results then in a slimmer looking outside. But the overall appearance is the same, just for this Yersinia pseudotuberculosis pseudotuberculosis one, we have an additional coil domain that stabilizes this upper part of the shell. What's nice and what is kind of interesting, especially for people interested in protein folding, is that we have a molecular knot that is found in all of those structures, where the N and the C termini come together at this position. So it's in the beginning, it's a bit difficult to see, but I hope you see that in a second, that you get here, this blue part, you know, go through here, and then we have this gold part here. This is the position of a molecular knot inside the structure. So probably when we look at it a bit more schematically, we see here, this is the N terminus coming here, goes all the way over here, it crosses and comes back here. So this is, I think, this is the largest protein for which a molecular knot has been described. Unfortunately, we don't know really the function of it, as for many other molecular knots. However, it could be important to stabilize this part where the linker goes out, and it could be important for the function of the protein. But this is how it nicely looks when you paint it with a pen. Yeah, so, but it's not only the periphery that's highly conserved with these different receptor binding domains, but also the center. And when we look inside, we see that we have a negatively charged pore that fits well when you isolate them. You can, there are, and they don't translocate proteins. They are cation permeable. So now to the BC component. This is of this Photorhabdus. We saw the structure of this part. It looks completely different. At this time, we used X-ray crystallography still. It forms a cocoon shape with which is like a huge barrel that is closed on the top and at the bottom. Here by a TCA binding domain, and up here by a hyperconserved domain. And when we look at this conserved domain up here, we see that it's a catalytic aspartyl protease site. So two of these aspartates, when we mutate them, we notice that the ADP ribosyl-transferase that sits inside the cocoon is not anymore cleaved. And therefore, we identified this pair as a catalytic diet that cleaves off its own tail, which resides then in the cocoon. And on the other side, we see that the cocoon is closed by this distorted beta propeller, where two plates are basically folded inside and blocking the central channel that's there, that would be there. So when you look from the side, we see this the cocoon with the gate up, down here is closed. We have the upper and the lower chamber, and inside the cocoon we have this high hydrophobicity that what we believe keeps the protein in an unfolding state. This was at least a prediction. To find out if this is really true, Daniel made a cross-linking mass spectrometry and showed that the ADP ribosyl-transferase that sits in there, although we don't have a density for it, is tumbling around and interacting with different regions inside the cocoon. So that would mean it's tumbling around, but it does not really say that it's not folded. And in order to find out this, we teamed up with a team of Hartmut Oschkinat's group in Berlin. And there they use solid state NMR and this what's called ultra-fast magic angle spinning NMR to determine if the protein inside is folded or not. And what they did, they used the cocoon once with the ADP ribosyl-transferase and once without. And these are the signals that one gets, the spectra. And then they compared it with the spectrum of the folded toxin. And you see that the folded toxin has these special signals out here. And they are not found when with the cocoon and the toxin. So that very much speaks for it that the protein in the cocoon is unfolded. Interestingly, recently we found a similar structure of this cocoon at an RHSA protein from type 6 secretion system. So in this case, this cocoon sits at the tip of this type 6 secretion needle and looks very similar to the cocoon from a Photorhabdus luminescens. But in this case as well, the protein inside is not folded, and so it seems to be a similar mechanism. However, in this case, the question is how this cocoon opens and how the protein that sits inside here, the toxin, is then released into the host. So this is still an open question that needs to be clarified in the future. So how do the two proteins go together? And this is what we call the holotoxin. So the BC component meets the A component, and this is a structure of this holotoxin complex, where this part adds to this part here. And during this, we see that a density that's non really defined enters the translocation channel down here. So how does that happen? When we look at this position, we see that the beta propeller that was before distorted at position 5 and 6 is now nicely ordered. It looks like a real beta propeller, and leaving a density here open, the cavity open. So when we look at the interface, we see that there is a clash with this loop of the TCA, and that would result basically in a restructuring of this part. However, this is just a morph basically, and if you look closely, there would a knot created during this. So this movement is biologically not possible. Therefore, we calculated MD simulations and found that one of these domains here, and then the other one, they completely unfold and fold back, but this time in the other conformation. So this is a refolding of a complex that goes through completely unfolding it, right, of two subdomains, which is, I think it's always very beautiful to see that, what biology can do. And what happens then is that the protein up here is then translocated through this channel, through this beta propeller into the translocation channel. The question is now, how does this protein enter the membrane? Again, now we are at this membrane protein center. So this is probably the most interesting part for you. And to find out how this works, we reconstituted the complex into liposomes and did negative stain and cryo-EM studies on it. And what we found is that the protein now sits on top of these liposomes and has a completely different structure. So before it looked like this bell, and now it looks like a little tree. And the only thing how this can happen must be that the shell opens and enters the membrane like this. So how is that possible? We identified an electrostatic lock at the bottom of the toxin that when you go to low and high pHs, that would create a opening of the channel and by repulsions, and would then make space for the sample part to go in. But how this worked, we did not know at the time, and how also this part works, that once it's inside its opening, we also did not know. And therefore, we embedded the TCA in the pore conformation in lipid nanodiscs. And now you can see we have a much higher resolution, and we see it's because the channel is open and it sits inside the membrane, right? What was kind of interesting is that the loops up here, so only until there we have really transmembrane helices, and on the bottom we have a loop. So how does this look in the membrane? We see that these the heads of the lipids in the membrane are intercalating between the protomers, especially at these loop regions, and stabilize this part of the complex. It's kind of interesting, so that we would have expected that the helices go all the way through and the loops are out there, but no, they sit inside the membrane. So then Daniel, in the very end, combined all of them and put it also in lipid nanodiscs. So we see now that also the cocoon fits into this cavity that's formed by the A component and enters the membrane. So this is very nice. And importantly, what we wanted to understand is now the conformational change from the prepore to the pore. So what happens inside the membrane, and when we see once this part is in the hydrophobic environment of the membrane, we have a change in the conformation. So a kinked helix is stretching out, becomes a full helix, and this then results in the opening of the channel at the bottom. You see that here again. On the left, there is a prepore, where on the bottom, the channel is closed. And at this point here, we have the opening once we are in the membrane of the lower part of the channel, and it results in the opening and a potential release of the protein that sits in here into the cytosol. So where does the energy for something like this come from? And for this, we compared between the prepore and pore. And we see that this linker domain here, that's very stretched, is compacted and partially folded in the pore conformation. To find out if this, compaction provides enough energy for this process, we did MD simulations of the isolated linker, where we measured that we had around 12 kilocalories per mole released by one linker when it goes from the stretched to the compacted form. And to find out if this is then enough to bring the protein into the membrane, we did coarse-grain MD simulations and studied the energy that's needed for TCA to enter the membrane. So you can see here it sits on top. You need a certain level, and this is a bit favored, but then you need this hill of activation energy to really bring this protein into the membrane. And once it sits then inside, which happens in a second, it's energetically favored, as it's expected from a hydrophobic tip. Yes, and when we calculate the energy between here and there, it's around 30 to 35 kilocalories per mole for a pentamer. So if one compares it P times thumb, the energy that's provided by the compaction of the linker should be sufficient for the protein to enter the membrane. So how long do I have actually?
We just a little bit over time. The presentation is 45. Yeah.
Okay, so I will summarize it quickly in like 5 minutes. So we also found glycans that acts as receptors for the TC toxins on the surface. So I'll go over that. And these are mostly Lewis X glycans. And what we showed, we connected them to BSA and proved that these glycans with the BSA bind to the periphery of the cocoon. And this basically shows that this Lewis X is interacting with the receptor binding domains on the surface. In addition, we also showed that heparins and heparan sulfates bind to TCA, but at this case, it was a bit surprising, at different positions, at different species. So, however, we have, in all of them, we have interactions with the shell surface, which brings us to this model that that heparins and other sugars recognize the complex on the surface, which results then in the endocytosis of the delta pH. So we have summarized it in a little cartoon. You can also find it on YouTube, where the BC component binds to the A component. This binding results in an opening of this beta propeller. Then the The ADP ribosyltransferase, which is unfolded here, gets translocated into the translocation channel here. The receptor, the TCA binds then on surface receptors that carry these glycans that specifically bind to the receptor binding domains here on the shell. This leads to the endocytosis of this toxin, and once it sits in this late endosome, we have and the endosome gets acidified, so the pH drops. We see that the color is changing. This simulates a pH drop. The shell here opens, and the translocation channel enters like a syringe into the membrane, releases the ADP ribosyltransferase into the cytosol. So I wanted, actually, this is, but it's all too long. We also have solved in the meantime the structure of this ADP ribosyltransferase, and it has been published in Nature Communications in the meantime. So this was Alex's work, and we also showed how this how the ADP ribosyltransferase interacts with actin. So I just go to the very end of it, and this is what we showed here as a summary, that we have this ADP ribosyltransferase that has a kind of a bridge mechanism, that once it binds to F-actin opens, so that NAD can bind and brings NAD close to threonine 148, which is modified by this ADP ribosylation reaction. And once ADP ribose sits on the F-actin, the protein here dissociates and cannot bind at the same position anymore. This binding of ADP ribose does not stabilize necessarily the F-actin filament, but it prevents the binding of cofilin and severing of the F-actin, and results therefore in an uncontrolled polymerization. So I want to thank all of the collaboration partners at different stages of this work, and I hope I have not forgotten anyone here. And I want to also thank my funding agencies and industrial collaboration partners, and I want to thank my group who did all the work. Thank you very much.
Yeah, thank you, Stefan. Thank you for the very interesting and great talk. So I guess it's the time we have, because we went a little bit over time, but I guess we can have some Q&A session as well, just for questions. So is there any questions from the audience? I see one questions in the chat. So from Edward, would you like to unmute yourself and ask your question?
Sure, I could try. I had a question about I noticed that you're talking about these receptor proteins by species and genus, and I was wondering if we're at the stage now where we can perhaps develop species or genus specific toxins, and that way maybe have a bit more control over some ecosystems without the damage caused by more generic and general toxins or pesticides. The same question would kind of affect, for example, I'm a parasitologist, and I know that there are a lot of protozoa and helminths that are not really well controlled. And I was wondering if also that could lead to an improved pharmacologic control of those.
Yes, thank you very much. It's a very good question. We try, and we have succeeded in some cases. However, it's relatively difficult to just remove or replace receptor binding domains with others without really decreasing the expressibility of the protein. So that's what happens. So somehow, I mean, one thinks this huge complexes, they express so easily in E. coli, but smaller modifications already render them non-expressible. And this is a bit of a problem. You know, when it's not just like a modular that you take something off and then it's fine. So most of the time, unfortunately, these receptor binding domains are also needed for stability of the complex. And this requires quite some investigations in the future. But in principle, it should be possible.
Okay, cool. Thank you. Is there any questions from audience? I think I can ask a question. So as I understood, so you, for example, for the TRPC, solving the structures of the, because you worked a lot on membrane protein, I just have a question on the technical side of your work. So that one, for example, you solved it in the detergent. So is there any preference of using, for example, detergent, nanodisc, or different lipid mimetic systems in your work?
Yeah, so we always try different ones. So with the trip channel, we have used the CDM CHS, but if we don't change to amphipoles, we don't get a beautiful single particle, for example, for recording. And in the case of calmodulin, for example, we change to element G because amphipoles they tend to cluster at high calcium concentrations. So we changed to element G. This worked well as well. In the case of the Ryanodine receptor, I think it was very beneficial to use lipid nanodisc, for example, because in this case, the protein is not really locked in a certain conformation. So this has been shown in the past that certain detergents or polymers have the tendency to lock the channel or the transporter in a certain conformation. And in this case, normally with a lipid nanodisc, this should not happen. And of course, you know, it's I think this is always the best. If nanodiscs work, I would always prefer this one. However, if the protein is small and the membrane protein, this can cause problems in aligning the images later for cryo-electron microscopy.
Okay, cool. Thank you. Is there any questions from the audience? So, yeah, I don't see any other questions, and I guess, yeah, we were a little bit over time as well. Okay, so I guess I will end this meeting, and I would like to thank you all for joining us today for this seminar, and a special thanks to our speaker who gave a very great and interesting talk. And I hope to see you all in the next seminar, which will be next week, from Dr. David Tall from Monash Institute of Pharmaceutical Science. Thank you so much. Have a great evening, everyone, and have a great rest of the day, Stefan.
Thank you.
Thank you.